So we will get started. And I'd like to say thank you to everyone for joining in today. My name is Dr. Elsa Zekeng, and welcome to this edition of our event, which is on unlocking the future of medicine with inclusive data and AI for drug discovery. So before I get started, I'd like to remind everyone it is a live event. So do with that information what you may. Please respect all housekeeping rules. Keep yourself on mute if you're not talking. Respect everyone's opinions, and please be open to learning, engaging, and sharing. So what is the background and the premise of this event? We ran a first edition last year, and that was very much on highlighting the role of diverse data in clinical trials. And so this year, we're taking the new theme on looking at how does inclusive data and AI play a role in drug discovery. And so we're thrilled to have you here as we explore the exciting intersection of AI, diverse data sets, as we know that medicine has to reflect all the population that we live in. And without this, it has led to many treatment gaps and disparities. But of course, in today's new era, there are lots of different opportunities. So over the next hour, we'll hear from leading experts who are pioneering in different frontiers, and we'll delve into the different ways in which we can engage with data sets and the role AI plays in it, and then its application, be it in the pharma industry as well as in the clinic space and directly to patients. To that end, I will start off by saying thank you again, and I'll go next introducing you to SökerData. So SökerData is a platform that I have the honor of founding and leading as CEO. And what we are doing is building an AI platform that allows us to be the trusted source of data from underrepresented groups, supporting safe and efficient drug design. What we get to do is accelerate access to correlated genomic, transcriptomic, and clinical data sets from underrepresented groups, so women and Black and ethnic minority communities, and we're starting off with breast cancer and cardiovascular diseases. I'm sure you'll be able to see the connection between what we're doing and, of course, the event we're holding today. So without further ado, I'd like to invite and bring on all our speakers, who will be great if them to introduce themselves. So I'd like to start off with Daniel Lowther.
Hi, everyone. So I'm Dan Lowther. I'm a clinical biomarker scientist looking to take medicines through clinical development pathway and try and get these ambitious medicines to patients. As far as background goes, I'm a wet lab scientist by training, but have taken and led teams in bioinformatics across biotech and pharma.
And I'll go next to Rory.
Hi, everyone. Nice to meet you, and thanks for the invitation, Elsa. I'm Rory. I'm a co-founder at a company called Unison. We have a data federation technology that enables you to analyze multi-omic data in disparate locations that is heterogeneous. And we have a machine learning and AI-driven harmonization tool that enables you to interoperate across those data for drug discovery, also clinical trial, patient stratification, etc. My background is in deep technology. I led research and development tech at Genomics England for 2.5 years. Prior to that, I was a consultant at Pfizer and led the COVID vaccine rollout strategy with the supply chain team there, and a long time ago, an academic in musculoskeletal sciences.
Thank you very much for that, Rory. And last and certainly not the least, Dr. Derek.
Hi there. My name is Dr. Derek. I'm a clinical oncologist. I work at the Beaton Cancer Centre in Glasgow. And I'm also the founder of an organization called Adopt a Doc, where we connect healthcare companies with healthcare professionals to empower them with the necessary critical insight. Lovely to be on this panel today.
Thank you. Okay, so we have about roughly 45 minutes to an hour to run through a couple of topics. And so we'll start off with building inclusive data sets, right? And I'd like this obviously to be as interactive as possible. And although I will start off going towards one of the panelists to speak towards a specific topic, if any of the other panelists you want to jump in and comment on it before the next question, by all means, please do. So the first thing that's been on my mind, and really part of why this event is holding, is how discussing strategies for creating diverse data sets for drug development. So I'll start off with you, Dan. I guess from a pharma perspective, what in your opinion or in your experience rather have been some of the key challenges in gathering data sets for drug discovery, and have there been any specific ones? So let's say engaging subpopulation or underrepresented populations.
Yeah, and it's a great question. I mean, one of the big challenges that comes with drug development is making the right decisions, and that's usually based on data. You want to be informed as best you can. Which patients do you enroll in your trials? Which patients do you target? Where is the opportunity going to be? Where is the unmet need? All of these are key things that we might think we know, but ultimately want to try and identify properly as we go through the process and learn from the trials that we do run. So building a data set and having a representative, and I'm going to use the word representative in this because, of course, you're trying to bring medicines to patients. So the patients that you're trying to derive benefit for, you need to have representative data for. And so if you've never included patients in your trial, how will you know that those are able to actually derive benefit? And does that mean you risk actually excluding patients from potential medicines because you don't have the evidence that it will work? So having any way you can build the right data sets in order to give you the confidence that either the trial is worth running because it gives a good probability of success. I mean, you don't want to develop a medicine that won't provide benefit to patients, or that you can take the learnings you've already generated and be able to provide access of this medicine to patients who maybe weren't able to enroll in the trial. And this can certainly be true for sort of minority individuals or diverse groups where in a sort of a couple of 100 patient trials, you can't possibly recreate the whole diversity of people. So especially across the globe, because you try and enroll many trials globally. And that can enable you to bring medicines to regions that maybe haven't got the infrastructure to set up, and maybe the academic centers needed. So that's probably the biggest use, is just actually get something that's, can really empower the data you've already generated and make the right decisions.
That's very powerful, and of course, there's so many different things that are crossing my mind. But I think I'll come to you, Rory, next, which is, again, considering the work that you've led and you've done in your past life, currently around with Unison, how can technology facilitate this collection of all these data sets that we're talking about? Obviously, Dan's just mentioned. Yeah, actually, let me pause there. Let me not ask a leading question. Let me not make it too leading.
Another great question. I think, as Dan was speaking, I was thinking about how we would be able to accelerate access to these data and actually extracting and utilizing them from a value perspective. So data federation is effectively an old technology, but hasn't really been utilized to the extent that it could be to add value. So hypothetically, if there were a hospital in developing markets underrepresented from a drug discovery perspective, we could create a federated architecture that integrates the data in that market to, let's say, big pharma or biotech companies' data internally. And then the bioinformaticians working with that data are working with it as if it were one sample. So I think federation could be part of the solution. I think also, there's an intersection of sort of digital and health tech, where by providing the opportunity to collect the data that's needed to support these decisions that Dan was talking about, and making that as easy as possible is really important. So that, for example, we're working with one big pharma, and a lot of the medical records they're bringing in from some countries are handwritten by clinicians still, and then scanned and uploaded. The reality is that's not very interpretable from a data analysis perspective. But using natural language processing, we can extract the pertinent data from those letters and integrate that into a database, which we could then integrate into some sort of, let's say, federated architecture that would enable you to analyze it alongside data that sits internally within the pharmaco, or from a clinical trial perspective, potentially across multiple centers in a multi-center clinical trial setup.
Very interesting. And of course, you just mentioned clinical. So of course, I want to come to you, Dr. Derek. From a clinical perspective, a couple of things that come to mind. Number one being, what's the direct application in patients, or at least let's say it was an impact in patients in terms of getting these data sets and actually implementing it in the discovery process from a pharmaceutical perspective. But also on the flip side to that, when you look at the tech perspective of what Rory was just saying, that how do we have clinical talk to tech, talk to pharma, right? And how do we use technology to merge all those lines?
I think that's a very good question. Well, data essentially is a representation of the realities that our patients go through. And I find it very interesting that what Rory mentioned about the federated data platform, because that is indeed how we can get all these different data sets that are currently missing. Back to the first question that you asked, like how do we deal with data sets that are missing in the clinical setting, and how do we get these diverse data sets from patients who traditionally do not engage with healthcare or doesn't engage with academic research? And that's something we certainly see a lot in our clinical trials. As quite a large cancer center, we run quite a lot of clinical trials. We try to get a lot of data sets to then inform our next clinical practice. But sometimes we find it really tricky, that's the word we use, to engage those who perhaps come from a more deprived socioeconomic background. And it's a lot of communication that's needed. There's a lot of communication that we need to take with them to explain why are we getting this data set, why are we needing these tools to build up these data sets, because we need to use this data to discover how better can we change our practice within the oncology setting. How can we evolve our service to not just treat the really well, rich and wealthy, but also the deprived people. And Glasgow is quite an interesting city where I'm from, because there are pockets of great wealth and also pockets of great deprivation. And oftentimes I think it's not even talking about the tech side of things, but it's really talking about how do you get the people to engage in the first place, because you can have as much platforms as you want, but if you don't have the people on the ground to actually convince the people to share their data and to actually sign up to certain programs, you're not going to get your tech, you're not going to get your data sets in the first place. Hope I'm making sense.
No, absolutely. And I guess then, which point, sticking to that data topic, if we come back to you, Dan, how are you solving that at the moment?
Yeah, that's a great question. We're trying to be creative. I mean, again, it's pulling the right. I would just sort of take a moment and go, it's all about working out what data you really need. So getting the right data is probably the most important thing, and that does come down to what's the question I'm trying to ask. So if you don't get the right question, then you won't know what data to grow and acquire, or the right way in which to acquire it. You know, especially when you're trying to integrate multiple data sets or bring them together. If you don't have the right kind of key information, it becomes very challenging to really learn from. So that's where we spend the most time. I mean, there is an abundance of data in the world, which is phenomenal, but that means you've got to be very sort of deliberate in the data you go after, and trying to make sure that it's the most impactful, the most relevant. And that again comes to, if we're thinking of the sort of clinical development path, a lot of the data that we base a lot of decisions on can be quite old. It can be sort of historic and has been collected, and does that reflect the actual situation that the patients have right now? Does it reflect the current standard of care? Or would they have seen, there's been a mass change in the sort of treatment landscapes in many diseases, especially oncology, with a lot of immunotherapies. That will fundamentally change how a patient responds to a therapy, how the tumor looks compared to the sort of the old standard chemotherapy, radiotherapy. So getting the most appropriate data will mean you'll be able to make an informed decision, or at least realistic findings.
Thanks, Dan. I know, Rory, you have your hand up. Please go for it.
Yeah, I agree with Dan. A couple of points on that. So firstly, it depends at what stage of the preclinical or clinical development pathway you're at in terms of what data you need, right? And there is an abundance of data, but the reality is we all believe that we've seen a hockey stick in terms of the amount of data generated in the last 5 to 10 years. That is a flat line compared to what's to come. If we consider multi-omics, other modalities, imaging, etc., deeper phenotype data. So there's a huge amount of value of being able to access all of those data and then apply, let's say, unsupervised machine learning models to it to identify, correlations or let's say clusters of interest that we currently are not perceiving as at present. I think then secondly, the reality is that there are underrepresented populations, and getting, for example, deep phenotype data plus omics genome, transcriptome, proteome, metabolome, plus imaging from those patients, like in the record entirely, at a sample size that's going to be significant from a statistical perspective, it's going to be hugely challenging. So I think there is a growing, let's say, prevalence of the use of synthetic data, and the way to approach that, I mean, any data scientist will tell me if I take a real world data set and then I create a statistically representative replica of that and multiply it by 10x so that I can train a machine learning model or do some bioinformatics on it, that I'm bonkers because the sample is inherently biased. But let's just suppose for a moment that we were able to create synthetic genomes, and we were able to introduce into those synthetic data pipelines the variance that's observed in the real world in different populations. So in fact, what you're then creating is not a statistically representative data set based on a small sample. You're introducing the variance that you perceive in the real world in different populations. And I think that's potentially a very interesting sort of way in which we might be able to address this disparity and this gap, and improve the efficacy and safety of some of the assets we're creating with the lack of data currently. And hopefully in the future, obviously, SökerData is going to solve this problem for all of us, and we'll have all of the data, the real world data. But anyway, I'll stop there.
Yeah.
Thanks, Rory. So, yeah, definitely.
No, I just think it's a very interesting point for that Rory mentioned about synthetic data, and I guess I don't really understand this space as much as some of the panelists as well. But I guess I do wonder if we develop an algorithm using synthetic data. I've not seen any regulatory approval given so far for any algorithms that's used for synthetic data thus far. But I can totally see the point that it can be used to generate the variance that we need. I guess the question is also, how do we generate the data in a sufficient variance to actually not be just one biased data set that's expand to grow a huge biased data set, instead of a biased data set that can just be grown to be a representative data set. Yeah.
So the way I would approach that, sorry to interject, Elsa, is you train a natural language processing model to look at the academic literature. So for the data that we do have in those populations, we understand the variance to some extent of the, let's say, the genome, protein, whatever the case may be, single cell, whatever the data or the metric is. And then you introduce that variance into the model. So you use historical data to introduce the variance, or you could do real world studies. Yeah. But I agree with you. I think that, you know, regulators are typically very good at what they do, but slow to catch up on what we're doing. And the reality is that you can use synthetic data now at a very early preclinical stage to get directional sort of directional insight, and then develop hypotheses that you can then go and test with real world data.
Fair enough.
So I guess, Dan, would you say that that's where sort of farmers is already hedging their bets?
Yeah, I was just thinking when Rory was talking, it's like, yeah, ultimately at some point you need to test it. But yes, having like, how do you incorporate it into your pipeline? Again, I come back to the point of you got to make good decisions, right? The likelihood of an early scientific idea making it into an actionable medicine is very small. So there is many examples of good medicines that fall off because they don't work in quite the way you think they would or in the real world. So how do you improve the likelihood of success of those? How do you pick the ones you want to place your bets on in the preclinical space? Because there's only a finite amount of resources you can take through. So yeah, you can incorporate it very much in an early stage, informing likelihood of success, how, with what's the unmet need, what are the patient populations we're going to go into, how do we see other competitive drugs performing? Do we believe that ours will differentiate? Or is there already a very good option out there for patients, in which case, let's look for another option where we can provide significant benefit. And then, yeah, ultimately, as you get through the pathway, can you speed up clinical development? Can you look for the right patient population as early? I mean, I'm coming from biomarkers. Can you find the biomarker early? Can you find the biomarker that will be able to predict response or explain what's going on, so that you can interpret the results faster? You can really insert it along the whole pipeline, and make sort of incremental sort of advantages throughout. But at some point, you've got to test it. That's again, at the moment, that's where the regulators sit. And that's the sort of the beauty of the A/B test in the clinical trial, is you've got to demonstrate it is better than what we currently have. But we can obviously make more informed and better better value bets.
Thanks, Dan. My last question actually on the data aspect, and actually I was just thinking about this to you, Dr. Derek. So there's a lot of data we've talked about, academic data, we've talked about proteomics, transformative genomics, like there's lots of throughout the process, but it still feels like there's still this gap in the translation into the clinical setting, right? Yeah. What are we missing? Is it that we're just all not talking to each other enough? Is it that we have a warped understanding of what is truly needed?
It's a fantastic question, and I think this itself can be an hour-long conversation. But I think, first of all, I think we should always start off by setting the scene that cancer by itself is incredibly complex, and we used to think it's going to be a one-hit hypothesis, one gene mutated, and then we have cancer. But we found out that's not the case. We realized that there's tumor suppression genes, there's epigenetics. We know that there's proteomics, there's the tumor microenvironment, there's the immune environment as well that helps grow the cancer. And we know that all these pathways have actually intertwined with each other and comes up to this very, very, very complex picture that the human brain is not very well equipped to actually manage this complexity. And I think Rory would understand the most, because with this multi-omics data, it's because we're trying to join all these lines together and try to figure out what's actually driving this cancer. And this complexity is why a drug that supposedly targets a pathway that's very well initiated, let's say that an ALK pathway for lung cancer, and we get an ALK drug. Why is it the response rate is only 30%, on 100% of patients who have ALK mutation, why only 30% of them actually respond to it? And really it's because there's this complexity that we're not perceiving from that point of view. And I know we're going to talk a bit more about personalized medicine later, but it's that complexity that prevents things from being translated to real life, because in real life we want to have a more simplistic, more easier way of doing things. We want to say, hey, this pathway we give this drug, that pathway we give that drug, when actually in real life everyone's a bit different. And with that complexity, it's difficult to execute in real life. How can we, for every single patient, analyze all this data that requires an entire data science team to figure out, okay, what works, what doesn't work? And the cost and resource is just untenable currently because it's just so expensive. But I think that's where the gap drops. There's a lot we can do in the research and academic setting, a lot of fancy things, in fact, because some very incredible stories to tell you. But those are not operationable in the clinical setting, where cost is a real concern, and there's thousands and thousands and thousands of patients keep coming through the pipeline. So that's my answer.
Thanks, Dr. Derek. Dan, yes, please go ahead.
No, I was just going to kind of really, what you're saying there really sparked a thought, which is, there's a very interesting paper that I'll put a plug in for, is how a biologist fixes a radio. It's worth having a read if anyone's got a spare sort of 5 minutes. It's like, we've got to remember this is biology, and biology is not engineering and physics, where we can't draw the electrical circuit diagram of a human body. We don't know exactly how each protein interacts with each other protein. Like when you read every scientific paper, everything is a balance, which means, an inhibitor, it's an inhibitor in one circumstance, but may well be a promoter in another one. You take some of the key genes we look at every day, and they form both roles. You hear, there'll be as many papers saying cytokines are pro-inflammatory as anti-inflammatory. So trying to dive through that complexity and find sort of empirical rules is really challenging. And that's where, but the only solution to that is getting more data so that we can understand what are common patterns, what are individual pieces, maybe individual patients. I mean, again, every person is different. The clinical response to someone who's grown up in London, who's maybe taken the tube every day and been exposed to all of the main pathogens on the tube and all that diversity, will be very different to someone who's grown up in sort of the rural countryside, where they may be seeing sort of animals around at the time. So the history of them, how they respond, is so different. We will only build this up over time, and for finding relationships that we don't necessarily empirically and sort of logically put together. It's just going to come through data.
Thank you very much, Dan. And I was laughing actually at, you said the diversity on the London tube. Anyone's been on the London tube, there's a lot of diversity on the there, but So this is a perfect, nice segue into the next section. So we're going into the application and the use of the data sets and how, when we layer on AI, as obviously you talked about trying to create those into an empirical formula, right? And which, coming to you, Rory, of course, you have quite a lot more experience around that whole aspect of application of technology, the usage of the data sets. Question being, how do we plug up the gaps that currently exist when we're trying to create such an empirical formula on something that is not like you said, it's biology, right? But we know that we don't have all the data sets. Obviously, Unison is creating our ability for us to have access to these data sets. But in the interim, how do we plug those gaps, and what role does AI play in being able to do that?
Another great question. You're knocking it out of the park. I think so. From my perspective, Unison doesn't provide access necessarily, but it helps you utilize data you do already have access to. Okay. So we're not in the business of selling access to data, sure, or being a broker in any way. What we want to do is to enable companies, organizations, or researchers in academia to be able to work across multiple data sets synchronously and, let's say, optimally, saving time from a data harmonization perspective. And the data harmonization really is the key point here. So you have multiple data sets in multiple jurisdictions. They typically have different data taxonomies, structures, ontologies. You can't just lift and shift those data into one location. If you were able to centralize them and automatically be able to utilize them and apply machine learning to try and identify some of these, let's say, interrelations between different mechanisms within the biological pathway. So I would say that the critical thing that we're coming up against now in single cell is that different technologies are used to process these data. There are different data structures. Every company's got a different way of doing things. There's no homogeneity. So when you're then given access as a big pharma or biotech to another single cell data set, it's not going to be interoperable with your own. So you then have to spend time interoperating the process. That's one of the problems we're really trying to solve with machine learning at Unison, is like how do we create a model that learns the different structures and ontologies of these different data sets and enables you to harmonize them. So data interoperability is the first thing. So just imagine if we did have single cell data, RNA-seq data, proteome, transcriptome, or excuse me, genome plus deep phenotype for a number of patients. Getting all of that data to be interoperable is the first challenge. Then looking at the gaps from a, let's say, sort of population representation is the next challenge. And I've been speaking to fairly senior academic in Singapore recently, and someone fairly senior within a big pharma company, about creating a statistically representative biobank globally. And I'd love SökerData to be a part of that, because it's one of the primary sort of, let's say, cabbaces that we need to address. If we can't fill that with real world data, then we need to look at other methodologies to be able to do it. And I believe that the regulators will require you to validate whatever technology you have in the real world, of course, through clinical trials with real patients today. But I think we can go some way to creating, let's say, confidence in our hypotheses through the use of synthetic data, as I alluded to earlier. I think gradually over time, one would hope that there will be a shift to more in silico-based clinical trials, especially at phase 1, which are higher risk for patients, so that we can develop higher confidence in the safety and efficacy of these assets before we take them into first and then I'll stop there at risk of rambling, but I hope it's...
Thank you so much, Rory. Very thorough. And of course, SökerData will be absolutely delighted to be part of this. I guess moving on to you, Dan. So, of course, everyone's been talking about pharma is going on to digital application of AI and all these different forms. Where would you say have been the best wins in terms of the application and some of the challenges that you faced in, sort of in terms of that application as well?
Yeah, so I'll sort of talk in theories and sort of concepts here, just to hopefully everyone can follow with the sort of where it's going. The AI brings an element of, again, this multidimensional data that we cannot possibly kind of sort through the complexity of. This was the days of that single biomarker being I found that one gene that predicts this one thing is largely over, primarily because there's enough people, very clever people looking for it, and you'll spot it very quickly. So the relationships and the complexity is what drives it, and then that is really going to be challenging to see and to prove. So using something like AI to pull together maybe connections that you wouldn't anticipate is often incredibly powerful. It's also, as you go into sort of the drug discovery modalities, where some of the ways that again, I talked about this sort of not electrical diagram, circuit diagrams, like pairing drugs, which work in two very different ways, but maybe very complementary. And like you would never put those two together and say, logically, for my phase 1, I'll put those two. So they're deriving a sort of a level of confidence that actually they hit two different pathways. They are complementary, or using the data that you've already generated, comparing it to the real world and seeing how representative that is, how likely are all these findings here going to translate into other populations is really important. It sort of makes data something that has an unlimited value. You can share it. It doesn't not only one copy. How you use it, you can is sort of maximize it. So I think it's really where I see the sort of benefit, and really trying to use technology to sort of add on to what a human does. We are very smart people that build on a lot of the literature that's been in there, but that's a set of experiments that have led to those data, and there is clearly a lot more to come. So yeah, really trying to make sure we're not biased in our own approaches to it and bringing our own preconceptions to it, but allowing the data to tell us what's going on rather than sort of us trying to tell the data what's going on.
Very interesting. And I guess, of course, I have to complete the triangle and come to you, Dr. Derek, which is, okay, we talked about it from a farm perspective. What would you like to see from a clinical perspective, right? If you had a whiteboard, and of course everyone is going, well, a blank check as well. What does that ideal look like?
So I can tell you a very interesting story, actually. And there's actually two very interesting stories that I've heard, and this is within Scotland itself. One of the very interesting things, and this is what Dan was mentioning as well, you talking about pairing drugs together. There was a professor in the Beatson Cancer Research Institute that took a patient's cancer and put it onto a platform and really tried different chemotherapy drugs on the cancer cell type and see what actually worked for this patient. And this was completely very experimental, very researching, and he can only do it for that one patient. And essentially, he found a particular combination for this drug, and it was a very random combination. We never seen like this pairing of drugs ever used in chemotherapy cancer patients, and you use it in the patient to some success, and that's a very interesting approach to myself. And there's also that big study, that big news recently about the Stanford professor who had brain cancer himself, and they actually took his genes and actually experimented on him, tried different drugs on him, and just chucked a new concoction to him, and he actually went into remission for one of the most aggressive cancer known to mankind, glioblastoma multiforme. And I guess I would like to see a lot more of this approach that is made easier and more scalable in a way that we can do this for more patients. It's just like, we for bacterial infection, what we do is we get a blood culture, we culture it in the lab, and then we grow it out. We grow the bacteria into a petri dish, and we try different antibiotics on it and see which area has the zone of clearance and say, oh yes, so this bacteria is sensitive to nitrofurantoin, gentamicin, amoxicillin. I kind of want to see that for cancer for every single patient, for them to be able to get their cancer cell, plate it up, check it through 10 different kinds of chemotherapy and say, hey, this works. This guy's cancer responds to this drug and this drug. Therefore, we should use this drug and this drug for him instead of, everyone must try carboplatin and paclitaxel. And because that's what works for the majority of people, and therefore we'll use it for everyone as well. So that one-size-fits-all way of doing things fits into our current model of cancer care delivery. That personalized, individualized, tailored approach doesn't fit, and probably would only fit for very, very rich and wealthy people, or very lucky people who have the professors to actually experiment on. So that's what I would like to see.
Right, thank you so much, Dr. Derek. That's actually two great stories. One which I obviously did not know of. So it's great to see. Interesting to see, is that paper, is it paper that's published? You know, the one where you said it diced up like the different one cancer and it had different trials of drugs. Is that published at all? Or if there's any
No,
links
that
to share?
that's an anecdotal. I've
Anecdotal
heard
story.
it directly from my professor as well. You know, it's mad science, but it's actually very, very interesting.
But also to your point, I mean, there are many sort of approaches using sort of organoids, sort of creating small versions of the tumors in the lab to be able to do screening for drugs and for drug combinations. Again, the key is just like taking that what seems like a very simple experiment, and it can yield a ton of data because you've got a whole culture of cells there. It's not just a single cell with a single drug. So different tumors have different kind of structures to them. And the different patients' tumors look different, have a different evolutionary history. So trying to dive through those extra dimensions as well with again, what seems like a very simple lab experiment to pull out the key information and turn something that is very complex and a kind of biological response into something that we can use in clinical practice, something that becomes very easy to understand, something that's highly predictive, because you don't want to sort of be able to say something like, well, we think this might work, but 50% of the time. Like it doesn't sound, you want to know as close to 100% as you can. And so how do you build that confidence and that, without having to literally just run it in the real world and then look retrospectively?
Rory?
Yeah, I agree. I think so. I've got a story. A good friend of mine's mother had quite an aggressive cancer. He did his PhD in genetics maybe 20 years ago, and has worked in various pharma roles and biotech roles. And he spent a lot of time trying to find a therapeutic to treat his mother, who sadly passed. I mean, she was also someone who I was quite close to. He took that approach and systematized it and developed effectively an algorithm for identifying repurposing opportunities. And they ended up acquiring an asset that failed phase 3 clinical trials about 10 years ago in prostate cancer and repurposing it for lymphoma, which they then divested of. And as a function of that, they're now raising money to, and hopefully working with us to build a machine learning algorithm that does that for individual patients and for specific therapeutics. So the first point is repurposing of assets that exist, potential combination therapies, and then also based on an individual's biological makeup, really, what therapeutics would they react most to? And there are differing opinions in the board in terms of the importance of each of those, but I like to think that the same algorithm in reality can be used in multiple ways to both in the clinic as sort of an oncology or oncologist support tool, but also from a repurposing and combination therapy perspective. So I think we're not far away from that. I think the data is obviously the critical component to this, and ensuring we have a sufficient data to train the algorithm, and that those data are easy to work with and don't contain any bias. And coming back to the diversity of the data and the importance of that, we'll be looking at ways in which we can introduce diversity into the training data sets that we work with that model.
Nicely brought on to the next topic I wanted to go into, and which is, of course, the biggest buzzword in the last however many months, AI, right? And I guess this question actually open to everyone. When we talk about AI data sets, of course, diversity in data sets, and in its application in this specific context, health, life sciences, and drug discovery, what are your biggest concerns? And this is the questions apply to everyone. So whoever would like to jump first, please go in and we'll roll off from there.
I mean, Mike, I'll go first. My big concern is you're going to get an answer. Is it the right answer? So, that's the one thing with this technology is, and with AI is like, depending on the quality of the data you put in, but you're going to get an answer that comes out the other end. It is going to predict something. It is going to find an association with something. And what I want to know and I want to have confidence in is how useful that information is. And so a lot of this actually comes down to the human putting the information in or also designing the algorithm to say, is it the right algorithm? Is it making the right trade-offs and the right balances in doing this? Is it weighing the data appropriately? Because I think any scientist will tell you they've seen algorithms that don't predict known biology at the end. You know, they sort of come up with this new cell type that we've never seen before in all of the years, because data associates in funny in certain ways, and if the data set is not representative, that can happen. Again, statistics isn't biology, so strength of p-values doesn't necessarily mean that two things are biologically related, especially when you get down to small numbers. So really making sure that it's designed appropriately, you got the right data in there, and then having a critical eye to say, does it give me the answer I actually want, as opposed to just an answer that I think I want.
Well, but then how do we innovate though, if we're looking for answers that we want, right? Because isn't innovation and isn't science discovering new frontiers? Maybe it's something that we don't know, right?
Completely. I think the key is just to make sure that it's almost not doing the opposite, right? That's why I talk about like known biology. You know, you don't sort of build on something where you can see from it that it's predicting things that are fundamentally, are fundamentally wrong. You know, like sort of the airplane that won't fly, or the boat that will sink. You know, so because that's where, especially when we take transcriptomics, when we mix it with genomics, there's a lot in there that we don't know, but there is also an awful lot we do. And so sort of taking that forwards is important, just to be the checks and balances. And then, if you see something, not, to your point, not assuming it won't work, but having some confidence that at least it's past the right triage points, so you've got some confidence.
Thanks, Dan.
My greatest worry, and I was thinking a lot about this over the past week, and I was listening to lots of podcasts and actually reflecting it on my own clinical practice. My worry is that we build an algorithm that not just build an algorithm from a data set that's very biased and that really benefits a certain group of people, but actually ignores or actively harm a separate group of individuals. And we see this a lot in real life. Let's put AI algorithms aside. We see this in the biases that we have as clinicians. We see this as the biases that the lack of knowledge and understanding that we have as clinicians, because we are treating someone that is a bit different or has different kinds of diseases or different kind of background. We presuppose certain things, we have our own prejudices, and then that person doesn't get the right treatment that's suitable for them. And we think that, that's real life and data is different, but actually data is really just a snapshot of the real world, And I worry that if we use data, even if it's a complete set of data, a complete snapshot of data, but it has within it the biases big inherent into it, I'm afraid that algorithms can be built that will systematically discriminate and actually actively harm patients who don't have actively harm patients who are not included in those data sets. And a very simple example I can give is this. So imagine right now we know that all the breast mammography data sets are mainly European in nature. All right. There's very little African and Asian breast mammography. That's just a fact. Globally, we don't have lots of Chinese, we don't have lots of Pakistani or Indian mammography data sets to work with. So a lot of these algorithms are built using European data sets. And we know certain races, they have a different density of the breast tissue, and certain races have more fat content. And what if an algorithm is fantastic at detecting that calcification in a European breast, but doesn't quite detect the breast calcification in an African breast? And what happens when algorithms say, yeah, this is all right, and this algorithm will have gotten the regulatory approval, FDA, MHRA approved, and operationalized in a medical center in, let's say, Edinburgh, or in London, and 10 European ladies going through, all of them all clear. An African lady goes through, they say, it's all clear as well, but the patient actually has a calcification in there, and they say, because different breasts can't really detect it. Patient say, oh, no breast cancer, you go home, and then the patient gets a breast cancer. So what do we? That's my greatest worry, that will happen. Obviously, we have to consider the needs of many, but it's these edge cases that makes or breaks a cancer, a healthcare setting, And you think that this is a hypothetical situation, but I have experienced something very real. I've experienced something very real. I have seen, but this is a human error. I've seen a patient completely, a locum breast consultant just completely misread the mammography, and a young lady with 2 young children had a cancer that could have been excised, but it's now metastasized. So I worry that that will happen with AI. So that's just my little fear.
Dr. Derek, do you want to come and for SökerData? You basically just said the premise of why we exist, so take you along on all of our pitches. Thank you for that. Rory?
Yeah, so a good friend of mine, Crystal Ruff, is head of AI compliance at Takeda in the US, and we often speak about some of the challenges associated with making sure you have the right data to train the model so that the outputs are then representative of the population or whatever the function of that model is supposed to be addressing. So I think from a compliance perspective, to Derek's point, as long as the model and whatever technology you're using has very clear guidelines in regard to the population that it has effectively been trained on from a data perspective, then I think we're okay. I think it would be a mistake, and it is a risk that you train these models on biased population data, and then they're applied in, let's say in scenarios in which those data are not representative of the population in which the model is being applied, or that particular function is being applied. That is a risk, and I think we need to be very acutely aware of that. And whenever we come across these tools, to really dig into the data that it was trained on. One would hope that in the not too distant future, there will be guidelines so that when you're releasing and launching these technologies in clinical settings. I've been mentoring startups now for about 7 years, probably 30 or 40 machine learning startups. Sadly, many of them have failed, but they all had clinical application. A few of them have gone on to be very successful. And they are very good at, when they're onboarding new users onto their technology, describing exactly what use cases the technology is very good at predicting and very good at addressing, and what it's not good at. I think as long as we're cognizant of that, and we all know to dig into the details and the data, we can kind of mitigate that risk to some extent, Derek. But I agree that it is a risk.
Sticking to you, Rory, first of all, thank you for that. But just sticking with you, because context, Rory will have to jump off shortly, but just to follow up as well on the point of its application of personalized medicine, right? What are some of the, again, from your experience, what are the low-hanging fruits that we get to have with what we have access to, and how we use and apply data sets, AI, machine learning technology in all these different user cases? And what would you say are the longer term things that we could potentially look towards and hope for?
So I really love this question, and something that I dwell on a lot. We had a newborn screening program at Genomics England. I think the initial ambition for that was 100,000 newborn genomes, but it's probably been reduced now to about 30,000. I met last week with Wendy Zhong from Harvard Medical School, who was really a pioneer of newborn screening. And it's a really exciting premise, right? You can screen a newborn, and you can identify whether it has, let's say, genetic variants associated with specific rare disease or cancer right now. But we're unable to actually take preventative action. And for me, preventative is the key word there, right? In the future, hopefully, we'll be able to develop these therapeutics, potentially even within hospitals. We're talking 10, 20 years out. I'm also speaking with the founder of In Silico Medicine, one of the world's leading AI drug development companies, Alex, about this. And obviously developing small molecules on site, because of the toxicity of it, probably not, let's say, viable unless we identify new technologies that can address that. But cellular gene therapies, why not? And I guess I beg a question to the audience, because I know there are some fairly senior people in here. Why haven't we been able to drive down the cost of cell and gene therapy drug development? I also work with gene therapy companies. One of my mentors and dear friends, Phil Beals, founded Exovia, has 3 assets and trying to treat rare sort of ocular conditions. If we get to a point in the future where we can drive down the cost and the efficiency of that manufacturing process, it's probably using AI to some extent, or machine learning. as well as the identification of specific biological mechanisms within newborns that are associated with disease. And those newborns we have very high confidence will experience a disease at some point in the future. I guess the future state is addressing that at the point of care before they leave the hospital, right? And if that's reality, then what needs to change from an industry perspective and a regulatory perspective? Because we're very used to treating chronic conditions over time and addressing illness with treatments over a duration, right? Or a period of time. There are gene therapies and cell therapies that are curative. If that was more broadly adaptive, what would that mean from an industry and regulatory perspective? And quite frankly, I don't have the answers to those questions, but it's something I'd like to dwell on. But I think that is, and it's a really exciting direction of travel.
Thank you so much, Rory. So I'll come to you, Dan, right? So when we're looking at personalized medicine within, again, with your pharma head on or with your pharma experience, etc., what are some of the points at which you feel that you're easily, some of the hurdles that you feel like you're easily climbing, and some of the hurdles that seem to be not insurmountable, but taking a bit more time? And of course, to Rory's question, well, the point that Rory was mentioning around the cost of cell and gene therapy, right? I mean, cell and gene therapy in general, but of course, the cost, we all know what some of those costs are, they're astronomical. And so there's that resource challenge that, of course, is happening. So yeah, what, with all of those factors in mind, those bits in mind, what are some of the small hurdles, insurmountable hurdles, or hurdles that you're looking at and go, something might have to change that's probably out of our remit at the moment?
Yeah, I think it's worth sort of thinking about when I think of personalized medicine, I sort of like it's right patient, right drug, right time, right dose. Kind of that's where trying to get all of that dimensionality together. So hurdles are everywhere. You know, can you actually classify the disease appropriately? Do you have a really actually good handle on the nature of that patient's disease? You know, again, the nature of this is that we tend to come with symptoms and presenting. We're not in that sort of really early stage, but we're trying. You know, you try screening early, especially with kind of circulating free or cell-free DNA, where you can get sort of liquid biopsy type tests and trying to sort of do early screening. That can have a massive impact. How translatable those results are to the real world is kind of key, and one thing we're developing data to understand better. So it's important making sort of decisions early, and then how well you've classified the problem, and then what tools you have in your armory to fix it, and how quickly you can do that. So there's this, I think the pharma industry will continue to generate a lot of very interesting products. There's almost a challenge out there to everybody else in the world, sort of like, especially drugs that are off patent that could be repurposed, of saying actually there is still utility in those, and it's, you know, it's where the incentive is to really find the right combination for the right person at the right time, that will make the biggest impact, not just to the disease but to the quality of life. I mean, it's certainly in oncology care that's almost as important a consideration as getting rid of the disease itself entirely. Again, so what do we mean by the word cure? Does it mean it's gone completely, or does it mean that actually we died of something else? So trying to sort of make sure we know what the end goal looks like, what does success look like, so that we can aim for it and try and achieve it, rather than setting unrealistic expectations and then sort of being disappointed that we fell short.
Just sticking to that, would you say there are any specific priority areas? And by that I mean in that whole pathway of, because it's such a complex challenge, right? So are you prioritizing any over the other, or yeah?
Yes, I mean it's the challenge, right? It's all about unmet need. So you know, with all of this pieces, it's you know, the companies, unfortunately, companies need to make money in order to survive and reinvest and keep going. So, but it's also, you know, it's all for the benefit of patients. So you know, that is at the heart of it all, and that's trying to be there. So where is the unmet need? Where can we add value and you know, help people ultimately? So trying to identify those are the right opportunities, because you're basically making especially for the pharma industry, you know, these things don't happen overnight. It takes a long time to develop a drug. It takes a lot of investment, a lot of people, like blood, sweat, and tears from individuals, and an awful lot of faith on the behalf of the patients to go with that journey with us. So, you want to have confidence that that's a journey worth taking, and these are efforts that pay off in the long term. They're not short-term, quick punts and easy ones. So, the more you can inform those decisions, the better. You can. You only got, as I said earlier, you only got so many hours, making sure your effort is the most impactful. Again, to individuals working, but also mainly to the patients.
Thank you so much, Dan. And then, of course, this is actually going to be my last question, and then I'll leave it open if you have any other things to mention. Would be when we're talking, of course, around personalized medicine. Of course, Dr. Derek, you shared around concerns around algorithmic really AI bias, the data sets being included. Do you have any successful examples, like any best case scenarios, best successful examples that you've seen in a clinical setting where it's all gone right? Or, I mean, you've obviously shared the ones about where it's all gone wrong. If you don't have any one where it's all gone right as well, that's fine.
Well, to be honest, I've not seen any specifically within the cancer setting. There are examples in other medical settings. There is, I think lots of you will probably know the standing together recommendations from University of Birmingham that talks about how do you get the 10 recommendations for diversity of data. How do you get diverse data sets? How do you build diverse data sets? So from that podcast, I was listening to a very specific example of an algorithm that assesses osteoarthritis. So it assesses osteoarthritis and determining when should a patient get a knee replacement or when they shouldn't get a knee replacement. And they realized that actually the way to mitigate a bias was not tuning the algorithm to weight the X-ray images more heavily, but actually to weight the symptoms of knee pain that's more heavily. So, and by doing that, they realized they actually mitigated the bias, and it actually made a more fair algorithm to actually make the right recommendations for different patients. So there are ways you could go about doing this, but I guess the challenge then comes is how do you identify in the first place that the algorithm is biased? How do you know it's not actually doing the right thing and saying, hey, actually that's not right, And how do you then know what to focus on? They were lucky because they found the right parameter to work on, which made the algorithm unbiased. But if you are talking about cancer, or talking about any other field where there's like 6 to 7 or 10 or 20 data inputs, how do you know which one do you need to play with to get to the right thing? So I think that those are the challenges. Nothing in cancer setting, but definitely good examples in healthcare in general.
Yeah, I think that's also where it's about leaving your ego at the door to some extent, because the whole scientific community, regards to discipline, has learned from each other. I'm going to quote a scientist, and I can't remember exactly. I wonder whether it was Newton, but it's like saying, like, I know I'm wrong, I just don't know how I'm wrong yet. So the key is, like, this is incremental. So we will all add more information to that pot to try and further this, but we can't get precious that we've got the answer, because the chances are we haven't got the answer. We've got an answer based on the information we have. So as long as we're constantly assessing it, constantly learning, then hopefully we can make continuous improvement. And I think that's the thing we should be focusing on, not to suddenly get 100% response rates in trials. If we do, phenomenal, but it's to build on and really make a big impact and get as close to that as we can, knowing that as over time we will get there.
Thank you so much, Dan. And on that beautiful note, I will end with, I like that quote, I know I'm wrong, but I just don't know how yet. And I think that's the most confidently humbling statement, which is still going in the direction of innovation, but still leaving room for error and not a new knowledge, not error, probably new knowledge. So I'd like to thank you so much for participating, and thank you for your time. Thank you to all who have joined and who will be rewatching this as well. And any further information, please reach out to each and every one of us or to myself. And without further ado, I will bring this to a close. So thank you again.
Thank you very much.
See you. Bye. Bye. Bye.